Hey, Assalamu alaikum. معكم دكتورة ياسمين أبوسمرة from Sonoran University, Arizona. Our topic for today is gluconeogenesis. This is very essential topic because without having this process, some of your cells cannot be able to survive. Okay, so today we're going to discuss what do we mean by the term gluconeogenesis, how our cells are going to synthesize glucose by this mechanism, and also when that process has to take place and where exactly, particularly in which part of the cell. So I assume that you already had a lecture of glycolysis, and glycolysis, of course, is utilization of glucose to release energy. Okay, so that's when we have plenty of glucose, whether you have a very good meal in the morning or during the day, and then you provide your circulation with the absorbed monosaccharides, which is particularly it's going to be glucose, then it enters glycolysis to provide your cell with energy. So whenever we talk about carbohydrates, we always link it to energy production. So that means when we take our food, of course, we don't think about it that way, okay? But we are triggered to ingest our food or take our nutrients because there is a need inside your system to take those molecules to provide the cells with energy. And it's all about energy, okay? So you cannot get to no energy in your cells and be alive. So we are doing so many mechanisms. We are taking our nutrients basically to satisfy the energy requirements. So this is why we are trying to substitute the lack or the low intake of carbohydrates by this mechanism. So let me move on and define gluconeogenesis. Okay, so this is a process that utilizes non-carbohydrates to make glucose. So this is the first thing that you should learn about gluconeogenesis, their definition, the definition of gluconeogenesis. And it's very simple, okay? I don't want you to memorize. I really want you to understand what do we mean specifically. So synthesis of glucose from non-carbohydrates. So if you are trying to synthesize glucose, that means that you are unable to take it from the diet, okay? So either we don't have access to carbohydrates, or people may decide not to take carbohydrates in their diet, like some nowadays people put themselves in keto diet, okay? So when they go for keto diet, that means they are taking basically proteins and lipids with very minimum amount of carbohydrates that might not be enough for certain cells to utilize. So the first condition is that we have drop in the carbohydrate levels, okay? And there should be a need for glucose or carbohydrates inside our cells. Then our cells will decide to synthesize glucose from non-carbohydrates. What do we mean by non-carbohydrates? So this is the part that I'm going to tell you in a second, because if we have carbohydrates, there is no need to synthesize glucose, and there is no need to do this process, which is gluconeogenesis. And then we're going to look at the key enzymes that are regulating this process and providing us with glucose. That glucose can be used by certain other organs or cells for energy, to satisfy the energy requirements. Okay, so when we define gluconeogenesis, we said that synthesis of glucose from non-carbohydrates. So non-carbohydrates are going to be our precursors. So how I can make glucose inside my cell without having carbohydrates. Okay, so this is very straightforward. And when we do any organic molecule synthesis, we are just focusing on the carbon molecules. We look at which molecules can provide me with the carbon atom. Okay, you know that glucose has 6 carbons. Now we need to make sure that we have the availability of those 6 carbons. If you have a good background in organic chemistry, when the synthetic molecules in the lab, for example, we can synthesize organic molecules if we can provide those carbons. We don't pay a lot of attention to hydrogens or oxygens, but we focus mainly on the carbons. So which non-carbohydrates molecules can provide me with those carbons? So we have some options. We have pyruvate. Pyruvate has 3 carbons. Lactate has 3 carbons. Glycerol also has 3 carbons. Amino acids, it depends on the type of the amino acids, whether they have R group that contains many substituents or not. But here we use the carbon skeleton part of the amino acid. So amino acid also can provide us with carbons. This is TCA cycle. This is Krebs cycle, also has different names. Intermediates here also, they can provide us with carbons. Okay, so if we can get the carbons from those molecules, then we should be able to synthesize glucose. Again, glucose has 6 carbons, and the rest are hydrogens and oxygen, and those are going to be the carbon donors. So again, this is in addition to what we said. In order to do gluconeogenesis in your cells, that means you have to be low in carbohydrates intake. So you don't take enough for many reasons, okay? Number 2, there should be a need for energy, okay? So you don't make glucose and just, eliminate it from your cells. You make glucose because you have certain cells that need that glucose to do their cellular activities and utilize the energy that's provided by that glucose. 3, which is this slide, we need to have a carbon donors. We need to have a precursor that can be used to synthesize glucose. Okay, next, I'm going to show you the details of how we can do this mechanism. So in any pathway, you have to learn those 4 concepts: when, where, why, and how. Okay? Now we know that we have to have certain conditions to do gluconeogenesis. In this slide, we're going to see how we are going to do gluconeogenesis. So in your previous lectures, I think that you learned how to get from glucose to pyruvate. So the process on the left-hand side is called glycolysis. So we take our glucose, phosphorylate it, convert it to fructose, and then phosphorylate it, continues until you split it into 2 pyruvates. Each one of them has 3 carbons, and this is the end of glycolysis. Of course, we produce some energy here because this is the main goal of glucose in our cells. Of course, it has some other functions, but this is the main goal of glucose, is to provide us with some energy. Okay, along this pathway, we produce energy in the form of ATP. We also produce NADH. This is also can be an energy source when it enters electron transport chain. Other possibility is when we have pyruvate. Pyruvate can be decarboxylated to acetyl-CoA, which enters TCA cycle to produce more energy. So the whole purpose of glucose is to get energy. This is why we are trying to go on the opposite direction to produce energy, to produce glucose, so we can send it to certain tissues to use it to produce energy. So please pay attention, because this point, one of the point of confusions, because you might ask, if I'm synthesizing glucose, okay, Is that mean the liver where, and by the way, gluconeogenesis takes place in the liver. Okay, liver is heavily synthesizing glucose. In addition to liver, also kidneys could do some gluconeogenesis, but we are going to focus on the liver. So liver will synthesize glucose, but it does not use that glucose for its own energy requirements. Once it's made, it will be released in the circulation, okay, and it will be sent to certain tissues such as brain, basically, and RBCs. Okay, brain prefers to use glucose. RBCs has no any other mechanism to use any other fuel molecules other than glucose, because RBCs do not have mitochondria. Okay, glucose can be, glycolysis takes place in the cytosol, so our RBCs are able to do glycolysis, but I mean lipid utilization or ketone body utilization requires a mitochondria, and our RBCs do not have that mitochondria. So it left us only option, which is glucose. So liver will happily make that glucose, send it to RBCs to be used, and liver does not use that glucose that they made. So let's come back again to energy versus glucose production. Gluconeogenesis requires energy. Okay, so as you can see here, the first step from converting pyruvate to glucose, we need energy. And we talked about low energy status of cells. So why are we making glucose? Because we need to do glycolysis to produce energy. Okay, so how come we should be able to synthesize glucose to release energy, while here the first step is still requiring energy? Okay, so this is the solution. Liver can get energy for itself. That could be from fat or lipids, okay, and provide or utilize that energy to synthesize glucose, okay, and this glucose can be used by other cells that needs energy. Okay, let's talk about just a comparison between brain and liver. For liver to be able to do gluconeogenesis, liver should have energy. But brain over there, brain does not have energy. So what brain needs? Brain needs this glucose to do glycolysis and get some energy. So we have two conditions here. Liver has energy, brain does not. Liver still has some means and ways to get that energy, brain does not. So brain is waiting for this glucose to do glycolysis and receive that energy. Okay, so from where a brain can get this glucose, it gets it from the liver, because the liver has all the enzymes, it has the ability to synthesize glucose and send it to the brain. Okay, so let's summarize it one more time. This is the major conditions. We said that we have low levels of carbohydrates in our circulation, including brain. If you don't take enough carbs, that means your brain is not receiving enough glucose. Okay, so brain is low in glucose. We need to fill that gap and send glucose to the brain, but we are not taking it from diet. Okay, liver will help. Okay, how liver will help? Liver will synthesize glucose and send it to the brain. So brain is low in energy. Liver has some other substituents, like can get energy from either stored energy or can get energy from fatty acid utilization. Okay, so now the conditions are ready for the liver. Liver has the pyruvate. Instead of this pyruvate, it could be any one of the substrates that I just showed you. It could be lactate, it could be glycerol, it could be carbon skeleton from the amino acids intermediates in the TCA cycle. So all these could provide carbons for this process. But the most immediate one is pyruvate, and I'm going to show you the mechanism by which liver should be able to synthesize glucose from pyruvate to send it to the cells and organs that they are needed. So it will start with pyruvate, and it has to go from pyruvate to phosphoenol pyruvate. The enzyme that converts the opposite reaction does not go, it's not reversible. Okay, this enzyme pyruvate kinase cannot go back and use pyruvate and convert it to phosphoenol pyruvate. So we call them bypasses. Okay, so we have to go from pyruvate to phosphoenol pyruvate in 2 steps. Okay, the first step is in the mitochondria. Okay, so remember, again, we have to be in the mitochondria. We have to do it in cells that they have mitochondria. So and liver cells, of course, or hepatocytes have those mitochondria. In the mitochondria, we have an enzyme called pyruvate carboxylase. Okay, so what does it do? It will add a carbon to pyruvate. Pyruvate has 3 carbons. Oxaloacetate has 4 carbons. This step and this enzyme requires ATP, requires energy. And luckily, liver should have and could be able to provide this ATP and do this process. And unfortunately, sometimes with liver diseases or deficiency of pyruvate carboxylase, the process of gluconeogenesis cannot occur. Okay, so let's continue in the normal way and under normal physiological conditions. Oxaloacetate will be released in another step by converted to another molecule, released into the circulation, into the cytosol, and there in the cytosol, oxaloacetate could be converted to phosphoenol pyruvate by an enzyme called phosphoenol pyruvate carboxy kinase, and this enzyme also requires energy. So this is another condition that you need to add it to, or factor, you need to add it to the process of gluconeogenesis, that it cannot start unless if we have energy. So high energy, low glucose content. That's an opposite of glycolysis. Glycolysis can take place when we have high glucose, low energy, because we use it to produce energy. Here, to synthesize glucose, we need energy, and we have to be at a low status. Glucose level is low, blood glucose level is low, then we synthesize glucose. Once we get phosphoenol pyruvate, we are going to use the glycolytic enzymes, the enzymes from glycolysis, because we are just reversing those steps. So we're going to go back from phosphoenol pyruvate to phosphoglycerate to 2 phosphoglycerate and then 3 phosphoglycerate, continue the reversible reactions and get 1,3-bis phosphoglycerate and then glyceraldehyde 3-phosphate until we get to fructose 1,6-bis phosphate. This enzyme here on the left-hand side, which is phosphofructokinase 1, PFK1 in glycolysis, it's an irreversible reaction. Okay, it has one direction. So we need to do another bypass and get to fructose 6-phosphate. And here we have another enzyme called fructose 1,6-bis phosphatase. Next, we use fructose 6-phosphate with the reversible enzyme here and form glucose 6-phosphate. Again, this is one of the 3 irreversible reactions in these pathways that needs a different enzyme, which is called glucose 6-phosphatase. This will remove the phosphate from glucose, and once glucose is free, then it can be transferred to the circulation, and it could be transported to brain as an example, or RBCs or some other organs. Okay, so a couple of things that we would like to mention here. For example, if somebody could ask me, why do we go from fructose 1,6-bis phosphate to fructose 6-phosphate, or why do we go from fructose 6-phosphate to fructose 1,6-bis phosphate in the same cell? Because those 2 reactions could happen in the same cell. Okay, what will be the driving force that we are going this way or that way? if you have fructose 6-phosphate, what determines the movement? Is it going, or the direction of the reaction? Is it going to be converted to glucose 6-phosphate, or it's going to continue on this direction? So the driving force here is the requirements by the cell. What do your cells need at this time when they are producing this fructose 6-phosphate, or when they are producing glucose 6-phosphate? So the direction of the reaction will be determined by normally energy status of the cell or the cellular requirements. So if the brain needs glucose, then this glucose 6-phosphate will continue to glucose, and by dephosphorylation, and glucose is a small molecule, then it could be transported to the brain cells and give them the energy that's needed. If we have glucose 6-phosphate and the liver cells requires energy, then glucose 6-phosphate will continue to glycolysis until it gets to pyruvate and give liver its energy, and this is called glycolysis. Gluconeogenesis is when we are going on the right hand side. So this is number one, okay? The home take message, the driving force, how we go from here to here, or how we go from here to here. It depends on the requirements. Your system is highly collaborative, okay? All organs are working together in harmony. Okay, and by harmony, that means they look at the requirements for each one, and they try to do their best to satisfy those energy requirements. Liver is the best organ that's dealing with all these issues in terms of gluconeogenesis, ketone body synthesis, and many other mechanisms. Like, I don't want to go into more details, like for example, we have bilirubin, for example, also toxins that could be handled within the liver. So it has so many mechanisms, and this is one of the major mechanisms that liver do for our cells. Another question is that how frequent do we do gluconeogenesis? Okay, so the straight answer, it's supposed to be during fasting state, okay, when we don't have enough carbohydrates intake, okay? So fasting state could be a condition. Some diseases could be a condition, or if people decide not to take carbohydrates, they are not in the fasting state, they are taking food, but it does not contain carbohydrates. Okay, so these are the general conditions when we do gluconeogenesis to synthesize that glucose and deliver it to the organs where it's supposed to be utilized. Okay, so what are the regulatory molecules? Which molecules that we are looking at when we talk about the regulation? Because we cannot do gluconeogenesis every single second in our lives, okay? Although there are so many researches determines that gluconeogenesis is a continuous process. Okay, so even if you are taking carbohydrates, still the liver will do a minimum amount of gluconeogenesis and producing very low amount of, it's not noticeable amount of glucose, just to make sure that the process is ongoing process and the enzymes are activated and ready to the fasting state. Okay, and we are on fed fasting cycle all day. Okay, we don't eat 24 hours, right, a day. We eat, we fast, we eat, we fast, especially at night, right? We fast for 8 to 12 hours at night. It depends on our feeding habits, but in general, this is the fasting time. So this is it on daily basis. We are be in a fasting state on daily basis. So the liver should be prepared to do gluconeogenesis, because this is the major source. Of course, we have glycogen degradation or glycogenolysis that provides some glucose, but gluconeogenesis is an effective mechanism by which we can maintain the blood glucose level. So that's why in certain researches, they said that it's a continuous process. It's not happening just in the fasted state, but also it happens during the fed state, but at a very minimum amount of glucose production. So this is something that you can just put it on the side just to understand the importance of this mechanism. Can you imagine if a cell cannot use any other fuels and cannot receive glucose? So those cells will die. And I'm talking basically here about the RBCs. Can we survive without red blood cells? So the answer is definitely no. So red blood cells will wait for the glucose to come and be utilized, whether we are in a fed state or fasted state or keto diet, or we're sick or we're healthy. RBCs always need that glucose. It has to get in there to provide them with the energy, to feed them with those fuels for their energy requirements. So the other question, is it important? Yes, it's important. Is it happening all the time? The question is yes and no, but we will say that if we would like to focus, or somebody ask you and give you multiple choice answers, like, is it happening all the time? Does it happen in fed state or fasted state? The priority is to consider it that's happening during the fasted state, but with a very minimum amount all day long, okay? And so we need to look at specific enzymes that are involved in this process from pyruvate to glucose. So the first enzyme is pyruvate carboxylase, and I said this enzyme requires some energy, and then we are going to use phosphoenol pyruvate carboxykinase. Those two enzymes is just to provide us with phosphoenol pyruvate from pyruvate. And we move on and get to the second one, which is fructose 1,6-bisphosphatase, and then finally glucose 6-phosphatase. And I'm going to show you here in each slide individually to see how those mechanisms are taking place into more details. And please, when you study biochemistry, please make sure that you learn your enzymes, okay? If you learn the enzymes and their cofactors and the regulatory molecules, you're done learning your biochemistry. So here from pyruvate, as you can see, the oxaloacetate with pyruvate carboxylase using energy in a form of ATP. This is happening in the mitochondria. And then the next step is from oxaloacetate to phosphoenol pyruvate by phosphoenol pyruvate carboxykinase enzyme, and requires energy in the form of GTP. Once we get to this point, which is now cytosolic reactions, we're going to continue on our reactions. So liver has this mechanism. Liver is able to synthesize glucose because liver has those two enzymes, particularly those two enzymes, and this is the starting point from pyruvate to glucose. But you can also see that lactate as a precursor can be first converted to pyruvate, and then we'll continue the same process. Amino acids, carbon skeleton, some of the carbon skeleton or some of the amino acids like alanine, by just removing the amino group from alanine, it will be converted to pyruvate. So we can use amino acid as an example, alanine as an example of amino acids to synthesize glucose. Okay, so if we are able to get to phosphoenol pyruvate, the second step that we need to bypass it is here from fructose 1,6-bisphosphate to fructose 6-phosphate, and this is dephosphorylation. We are removing one of the phosphate molecules, and the enzyme here is fructose 1,6-bisphosphatase. And when you see the term phosphatase, that means we are removing a phosphate. If it's kinase, then we are adding a phosphate. So phosphatase, here we have two phosphatases that removes a phosphate molecule from fructose 1,6-bisphosphate. Next, we're going to get to glucose 6-phosphate, and now we need to remove this phosphate to make it free glucose before it can be transported in the circulation. So what do we need? We need another phosphatase. Again, the same thing, phosphatase enzyme to remove this phosphate from the glucose, and the enzyme here called glucose 6-phosphatase, and I just put it here. Okay, so these are the three reactions and the most regulatory reactions within the process of gluconeogenesis. They are regulated enzymes. Some of them have cofactors that cannot work without them. But again, the direction of the reaction is determined by the status of the cell, of the cells themselves. Okay, so we could ask a question, for example, why some other organs are unable to synthesize glucose by gluconeogenesis and maintain the blood glucose level. Example here, like muscles. Muscles do not have this enzyme. Glucose 6-phosphatase is missing. Muscles do not have it at all. So there is no point to do gluconeogenesis, synthesize glucose, and try to send it somewhere else to be used, okay, to be used in there for or to produce energy, because there is no way that we can get this free glucose from muscles, because they don't have glucose 6-phosphatases. Those reactions are taking place in the cytosol, but specifically in endoplasmic reticulum. Okay, so we talked about when do we need to do it. So the conditions like low carbohydrate intake, we have to have some energy in the liver, at least the minimum amount of energy in the liver to start the process. But we have to have some tissues that waiting for that glucose to produce energy. Okay, we need to make sure that all the enzymes are active and available. Okay, like we know that we have those enzymes in the liver, but we don't have them in some other organs like in muscles. Okay, so besides that, of course, we need to think about the hormonal regulation. The metabolic pathways are generally, specifically if we're talking about carbohydrate metabolic regulations or metabolism, we should pay attention to which hormones are released or secreted and which hormones are not. In the case of gluconeogenesis, insulin will drop because insulin is released or secreted when we have carbohydrate intake, and it drops when we don't have carbohydrates. So which enzymes are raised here is hormones. We have glucagon. Okay, in this condition, glucagon is the regulatory hormone for gluconeogenesis because it will increase and stimulate the process. Also, when pyruvate will be converted to oxaloacetate in the first step, it will be stimulated and inhibited by certain molecules. Okay, so the first enzyme in the reaction requires energy, but also it requires biotin. And this is just something that to remember. When we see biotin, that means there is always, most of the time, there is a carboxylation reaction, addition of carbon. Addition of carbon in general requires an enzyme and also a coenzyme. Coenzyme is like a helper, assistant to the enzyme. So in this case, we have biotin, add the carbon atom to pyruvate to form the oxaloacetate. The only difference between pyruvate and oxaloacetate is the number of carbons. Here, pyruvate 3 carbons, oxaloacetate is 4 carbons. And when I say the difference, I mean from structural point of view, and specifically I'm focusing on the number of carbons. So when ATP or acetyl-CoA are high, we need to be, the liver has to have this energy, and if not, liver cannot do gluconeogenesis. Okay, but once we have high level of ATP or acetyl-CoA, once we have acetyl-CoA, that means it could enter TCA cycle to produce energy, then pyruvate can start gluconeogenesis. Otherwise, if we don't have acetyl-CoA, pyruvate will continue decarboxylation reaction to produce acetyl-CoA to produce more energy. So the priority is always for energy, because liver, if liver does not have energy, it cannot synthesize it. It cannot help others. I mean, if you are unable to help yourself, you cannot help others. So that's the rule. So liver will, first of all, has to be in a high energy status to be able to use that energy to synthesize gluconeogenesis, to glucose by gluconeogenesis. We have some other factors and some other molecules that will help you to understand the status of the energy and how the enzymes will be activated. So one of the steps that we need to hydrolyze F1,6-bisphosphate, okay, and basically by hydrolyzing it here, we means that we need to remove one of the two phosphates to get to fructose 6-phosphate. This requires an enzyme, and we talked about it. It's a phosphatase enzyme, and this enzyme is regulated by 2 or 3 conditions. The first one, the citrate will stimulate it. Citrate is associated with high energy status. If you study, I'm not sure if you already studied TCA cycle, this is the first molecule that's synthesized in the TCA cycle, and TCA cycle is a cycle or a pathway that produces energy. If we have citrate, that means we can get energy and the gluconeogenesis will be stimulated, and this is one of the enzymes of gluconeogenesis will be also stimulated. I'm going to jump to AMP inhibits. AMP is an indication of low energy status. If we have low energy status, liver cannot do gluconeogenesis, so it will be inhibited. Okay, this is a very interesting molecule, fructose 2,6-bisphosphate. It says here it inhibits gluconeogenesis. It inhibits glucose synthesis. So why? What's the correlation between this molecule and gluconeogenesis? Number one, fructose 2,6-bisphosphate is an intermediate in a carbohydrate metabolic pathway where we should have high amount of carbohydrates to convert fructose to fructose 2,6-bisphosphate. So this is an indication of having a high amount of glucose or carbohydrates. So if we have carbohydrates, there is no need to synthesize glucose. So that's why a molecule like this will inhibit the process of gluconeogenesis, and in fact this molecule is an allosteric regulator for fructose bisphosphate PKF enzyme that's needed for glycolysis. Okay, so this molecule will activate glycolysis. That means we have glucose, we have carbohydrates, so that means we don't need to synthesize glucose and it will be inhibited. So these are just examples of the most important regulatory molecules that will either inhibit or stimulate the process of gluconeogenesis. And please try to understand the concept. Don't try to memorize the molecules. So the main concepts, anything that provides energy will stimulate gluconeogenesis. Anything that shows you that there is carbohydrates in your cells, this will inhibit gluconeogenesis. Okay, so glucose level inhibits gluconeogenesis, energy level stimulates gluconeogenesis, and these are the two major umbrellas where you should be able to answer any question if you know those two concepts, and you can just apply all other molecules as regulatory molecules. Okay, so another enzyme that is very essential enzyme, it's well studied and it has clinical concerns if it's deficient, which is called glucose 6-phosphatase. Okay, so glucose 6-phosphatase converts glucose 6-phosphate to glucose. Just remove the phosphate from glucose. And I said this is very essential because you need to get free glucose to circulation. We found it in liver, we found it in kidneys, so that's why they are able, excuse me. So they are able to synthesize glucose and they have that mechanism, and they should be fine to synthesize glucose and send it to where it's needed. Muscles and brain, they don't have that enzyme and they don't do gluconeogenesis, and glucose 6-phosphate is under substrate level control, meaning that if we have glucose 6-phosphate, then it will be activated based on the other conditions and give us those products at this level and be able to have that free glucose, and it will be regulated by the availability of glucose 6-phosphate or the absence of glucose 6-phosphate. Okay, so this slide will show you the details about the molecules that I just talked about, like fructose 2,6-bisphosphate. So it's negatively regulating gluconeogenesis. That means it will inhibit it. But if you look on the other side, on glycolysis, it will activate PFK over here. Okay, so it will activate the process of glycolysis, and that's an indication of what? If activating glycolysis, that means we do have carbohydrates. So that's why it's an inhibitor of gluconeogenesis, because whenever we produce it, whenever our cells are synthesizing this molecule, it's a great indication of presence of carbohydrates or presence of glucose. So activates glycolysis, inhibits gluconeogenesis. And you're going to see in some molecules like AMP here or ATP here or citrate here and so on and so forth. But all of them are indication of either availability of glucose or absence of glucose, availability of energy, or low energy status. We don't say absence of energy because there is no cell that could tolerate or survive without a minimal amount of energy. Okay, so this is a question for you just when you go through these slides. Just pause a little bit and think about what is the specific role of fructose 2,6-bisphosphate in glucose metabolism. Okay, so whether you're doing glycolysis or whether you are doing gluconeogenesis, and try to make sure that you truly understand the role of this molecule, and then you should be able to understand how regulation of these two mechanisms are done by different molecules. This is just an example. You can remove this one and put citrate. You can remove this one and put ATP or AMP or any other molecule, and then you can apply the two concepts, energy and glucose availability. Okay, so finally, I just want to show you what will happen if we are unable to do gluconeogenesis. So as I mentioned, it's a continuous process. We can see highly active during fasting state, but what will happen if we are unable to do gluconeogenesis, and why we might not be able to do gluconeogenesis? There are certain conditions that we might not be able to do gluconeogenesis. For example, if we are producing high levels of insulin, insulin secreting tumors, that will lead to hypoglycemia and will start to affect and negatively affecting gluconeogenesis. So things like if the insulin level is not stable, okay, or produced in high quantities, if we produce high quantities of insulin, this will give a wrong indication to the cell that there is carbohydrates available, but they are not. Okay, so this will prevent glycogenolysis, degradation of glycogen, or gluconeogenesis. And in both two cases here, we are trying to have glucose. But if we see that there is high level of insulin, the insulin is indication of high carbohydrates. So why are we able to do these two pathways while our cells is telling us there is carbohydrates? So that's a wrong message, okay? And insulin will act on those two mechanisms and inhibit both of them. Another thing, alcohol. Alcohol, high levels of alcohol will also lead to the same condition, which is hypoglycemia. Hypoglycemia means low blood glucose level, and in these processes, the ratio between NAD and NADH is very essential. And when people ingesting or taking alcohols, alcohol metabolism will release high amounts of NADHs, okay, and utilize NAD. And this molecule and this ratio is very essential for cellular activities and mechanisms. And one of them that's highly affected by this ratio is gluconeogenesis. So this will inhibit gluconeogenesis, alcohol consumption, overproduction of insulin, liver diseases or tumors, and also there are some mutations with certain enzymes. So if we are trying to make a process or a pathway, we have to make sure that the enzymes are active. The enzymes that are doing the process, doing the pathway itself, they should be active. Sometimes we are talking about enzyme induction, so we need to synthesize them from scratch. But enzymes here for gluconeogenesis, they exist, and they just need to be activated to do the process. So if any of those conditions applied to our cells, this will result into inhibition of gluconeogenesis, meaning that we are not able to have enough glucose to maintain the normal blood glucose level, and this condition will result into what we call it hypoglycemia, or low blood sugars, low glucose level is considered or known as hypoglycemia, and this is one of the major conditions that are clinically correlates to gluconeogenesis. Okay, any questions, please? I'm trying to stop sharing now.